Our next session is really exciting. When it comes to rare cancers, it's kind of a whole different ballgame. It's great to be rare if you're a car, maybe a gem, maybe a great bottle of wine. But when you're rare because of your cancer, it's really hard. And it creates a lot of anguish. And that's because you feel so alone and you feel like you're an N of one. And your physicians are telling you they don't have the data and they don't have the research to help you as much as they would like. So that's why it gives me such great pleasure to introduce my colleague, Joan Levy, who is our Chief Science Officer here at MRA, and a couple of other colleagues in the field who are working really, really hard to aggregate that data, do deeper research, find answers, find treatment solutions that are addressing needs that really haven't been worked out yet enough to help all of us. So with that, I'm going to introduce Dr. Joan Levy, Dr. Vito Rebecca from Johns Hopkins, Dr. Kasey from the University of Colorado, and Dr. Keiran Smalley at Moffitt Cancer Center.
Okay, thank you. I just want to say that Wednesday is a really busy day for all of us because 2 doors down, we've been in a grant review committee meeting since 8 o'clock in the morning, trying to identify the best research proposals to fund, and I've been in there, but I was so thrilled that Dana and Renee invited me to moderate this panel because it means a lot for me to be with all of you at the forum and be given the opportunity to run a panel and step a little away from the Grant Review Committee. I think you guys are more exciting. So I just want to start and just introduce the rare melanoma subtypes that we're going to be talking about today. Actually, this year, about 104,000 cases are going to be diagnosed of melanoma in the United States. 90% of those cases are cutaneous, or the more common form of melanoma. And then the remaining 10% are composed of 3 rare subtypes, and they include acral melanoma, which occurs on the palms of the hands, the soles of the feet, and the nail beds. Mucosal melanoma, which is diagnosed on the mucosal linings of various tissues and organs, and then uveal melanoma, which is melanoma of the eye. And as Dana pointed out, really, these rare melanoma subtypes, they're typically diagnosed at a much more advanced stage of disease. They could have, therefore, a poorer prognosis. And the approved therapies, these 17 approved therapies, except for 2, which are more geared towards uveal melanoma, generally are not as effective in treating these rare subtypes, more so than cutaneous. So you might know that MRA is the largest private funder of melanoma research worldwide. Before I get to this slide, we've supported, since 2007, $164 million in funding to support greater than 485 grants worldwide. And from the very beginning, we noted the importance of providing funding to rare melanoma research. So out of that $164 million, we've funded about $20.5 million to support 56 rare melanoma projects through our different grant mechanisms. So what have we learned from that funding? And not only from us, but from other sources of funding. Well, first of all, when you look at the different subtypes of melanoma, including the most common cutaneous and the 3 rare subtypes, you understand what's called its genomics better. So we help support research that did genomic sequencing of different tumor tissues, and this helped identify genomic alterations in the tumors, We really call them tumor biomarkers. And as you could see, if you look at each different color as a different genomic alteration or biomarker, you see that they're very different in, there are some common ones, but the numbers of the different mutations and types of mutations are different among the different subtypes. And then if you go on from that understanding, you're able to identify certain targets. What's a target? A target is a protein that we use to identify drugs against and help develop drugs. And from our research studies, as well as others, we've been able to identify targets in the various types of melanomas. The listing here is more specific for acral melanoma, but we've been able to identify targets for all the rare subtypes. But then researchers came to us and said, I don't know, I have these drugs to test against these targets, but I don't have these preclinical models to test them in. So where can I find these, for rare melanoma subtypes, so where can I find these models? So, and that would be like testing in cell lines and testing in mouse models. So what we did was we created this virtual catalog on our website to help connect researchers who have these rare preclinical models to researchers who need them. And we'll probably be talking about these preclinical models during the panel discussion. And then finally, we said, how can we really advance rare melanoma research further? So several years ago, and I'll talk about this a little further, we started the rare registry specifically for patients with acral and mucosal melanoma. And I want to say that it's a direct-to-patient registry, and I'm going to give you more information about it now to share. So how were we inspired to do this registry? Well, the registry started when a group of patient advocates came to us and said, we really want more research done for rare melanomas. And we really feel that researchers and even clinicians need to know more about our disease journey. So what we thought with MRA's help would be for you to be able to help sponsor this direct-to-patient registry. So I first want to say that these patient advisors were really, really inspirational. And I know a number of them are in the audience. So I just want, if they're not too embarrassed to do so, to have them stand, because I really think they earn recognition of helping us and inspiring us. So I see some of them out there, and if you could all stand. And thank you for all your support. I want to say you might be wondering now why uveal melanoma, another rare subtype, is not included. And that's because there are several other registries that are focused on uveal melanoma, including one from a Keiran site. And they have a uveal melanoma registry, and we didn't want to be competitive with them, so we wanted to be collaborative. And so we didn't want to include uveal patients because we knew they were being studied. So anyway, the patient advisors were joined by a group of medical and scientific advisors who really helped drive this registry forward. So what is this rare, it's called RARE registry, and it has become a signature program for the MRA. It is a direct-to-patient registry. I'll get into it more in a minute. It really brings together patients, caregivers, researchers, and clinicians. It's a community. We're all working towards a goal of accelerating research focused on rare melanoma subtypes. And as I just mentioned, it was built by patients, and it's for patients. And that's why we're so proud of it. So the RARE registry is a web-based direct-to-patient registry, and it's for acral, mucosal, and we've also included cutaneous melanoma patients. And what you do is when you join the registry, and you have to consent to join, you fill out a series of surveys, and you're also able to upload medical reports. So what do participants really get out of this? Well, what they have is they have these dashboards, and what they're able to get out of it is they're able to see accumulating real-time aggregated information on their rare subtypes. And everything's de-identified, which means you can't identify a patient in it by name or address or any personal identifiable information. And then what they get in return, so they can see this information on the dashboards. They could actually click on a question they answered and see how other participants answered that question, so where they stand relative to other participants. They get clinical trial alerts, webinar notifications. So they're really, really connected. And for researchers, we're building a research portal to share this de-identified aggregated data with researchers so that they could really learn more about the patient's journey, treatment responses, and what are the true needs for rare melanoma subtypes. So what's coming soon in terms of addition to the registry, we really want more clinical information of the participants. So participants will be able, if they consent to be able to dial into their electronic medical records. And if they give us permission, they could download their medical EHR information onto the portal, and then we could abstract additional clinical information. We're building an MRA melanoma biorepository to allow participants to collect tissue and provide it to the biorepository so we can conduct more research. And then, of course, as I mentioned, we're building a research portal for researchers to access this de-identified, patient-reported, as well as clinically integrated information to advance research for these rare melanoma subtypes. So at this point, I'll stop here. If anyone wants to learn more about the rare registry, please see me. There's information outside. Renee and Dana have information. But really now it's my pleasure to join the panelists, Dr. Kasey Couts from the University of Colorado, Dr. Vito Rebecca from Johns Hopkins, and Dr. Keiran Smalley from Moffitt Cancer Center to have this panel discussion. Seem far away.
Yeah, close enough.
Okay, so what I'm going to start off with is ask each of you for like 2 to 3 minutes to spend some time on really talking about your area of expertise and what your research is really focused on, really at a high level. And Vito, would you like to start?
Sure. Thank you, Joan, and thank you, MRA, for everything that you've made possible for rare melanoma research. I just wanted to first begin by saying thank you to all of you. As the son of a mother who passed from cancer, I've witnessed firsthand how devastating this disease can be. And many of you have experienced firsthand how devastating this disease is. And so I just want all of you to know that you are our motivation, our inspiration, the source of the passion for our work. So I just hope you know how special you are to the community. So just that. So my laboratory at Johns Hopkins, we focus exclusively on acral melanoma. Many of the aspects of this disease remain enigmatic. It's a complete mystery. We still don't know what causes the initiation of acral. This has escaped us. There's some common sense educated guesses that since acral is most common on the bottom of the foot, surely maybe mechanical forces of walking might be involved, maybe injury. But again, these are educated guesses that have yet been experimentally explored. So this is an area that our team is actively looking into. In terms of what allows this tumor cell to escape the foot, which is the most common site, and metastasize to the liver, the lung, the brain, this remains also a mystery. There's a few papers here and there that have tried to study it. If you look at cutaneous melanoma literature, there's more than 5,000 papers documenting mechanisms of metastasis. If you look at acral melanoma, there's less than 10. So there's such little that's been done so far. And obviously, this is a massive gap that, with the help of the MRA, needs to be filled because it's metastatic disease that we treat patients for. Another area our lab focuses on is trying to understand why existing therapies that have been approved and studied in the laboratory for cutaneous melanoma, given to acral patients, because acral melanoma patients, there's no current FDA-approved treatment specifically for acral melanoma patients. And although many acral melanoma patients benefit from approved cutaneous therapies like immunotherapy and targeted therapy, they don't experience the same benefits. And the reasons this is so have not really been explored either. So our lab is also trying to understand the resistance mechanisms that are preventing the acral melanoma patient population from experiencing the same clinical benefits that their cutaneous melanoma counterparts do, and trying to develop tailored therapy specifically for acral melanoma biology so that we can get those patients to experience those same clinical benefits, if not more. And so that's kind of just the high level of my work.
Thank you so much. Kasey?
Yeah, so thanks, Joan and the MRA, for the opportunity to be here and really not just share with you what we're doing on the rare melanoma research front, but also to get your feedback and your input and your thoughts on the rare melanoma research field and what we should be doing, where we should be going. So I hope a lot of you have some good questions and comments at the session at the end of the panel today. So at Colorado, really the heart and soul of our melanoma research program is a skin cancer biorepository that was started 20 years ago now, back in 2005 by Dr. Bill Robinson. So basically every patient that comes through the melanoma clinic, hopefully we try and consent the patient, we collect tissue, we collect clinical data. A lot of these tissues we injected into mice to make melanoma PDX models is what we call them, and we have about 175 melanoma PDX models now. But what really led us to the rare melanoma research field and my specific interest in mucosal melanoma is just that over time we accumulated a number of these rare melanoma specimens. And so as Joan pointed out in her first slide, mucosal melanoma really is the rarest of the rare melanomas. It's only about 1% of all cases. And within those, Joan also noted the different anatomic diversity of mucosal melanoma is so much greater than the other subtypes. So we have mucosal melanomas in the mouth, in the gums, on the hard palate of the mouth. We have it in the nasal cavity. We have it in the inner ear canal. We have it in the sinus canal. We have it in the vagina, in the vulvar regions, in the anorectal regions. And so it's really, besides just being the rarest of the rare, within this 1% is really several different cancers within that 1% of mucosal melanoma. And so over time, we've collected a number of samples of mucosal melanoma, patient tumors, as well as developed, we have about, I think, 30 PDX models now for mucosal melanoma representing vaginal melanoma, nasal melanoma, and rectal melanoma. And so really a big part of my research is to, one, utilize these samples to really better understand these anatomically diverse mucosal melanomas in terms of both their genomic profile as well as the different environments. So the tumor microenvironments are hugely different in these tissues. So we've been looking, for example, at the microbiome. So the tumor microbiome in the vagina versus the nasal canal versus the rectum is hugely different. And how does that impact the tumors? And so another area is just really trying to really build the collaborative community. And so over time, this biorepository, we have a really open sharing protocol and collaboration protocol. So we've sent specimens around the world from the biorepository. There's now over 220 publications that have resulted from the sharing of these samples. And just since Joan and the MRAs launched the catalog, the MRA catalog for these preclinical models back in 2021, we've actually shared now cell lines and PDX models with over 40 labs in the past 4 years. So really a big part of what we're doing is really trying to understand these anatomically diverse mucosal melanomas, because we know that they're very different and they shouldn't really be treated the same. They have different responses to immunotherapies, different targeted therapy indications. But we're really also trying to help just build the community and really build the collaboration. And the MRA has been a really huge part of sort of facilitating that role to help put researchers in touch with one another and really help share what's available and what's ongoing.
Yeah, thank you, Kasey. I think you brought up a really great point that we're going to go into further in a few minutes is, we're going to have to study these rare subtypes. collaboratively, because it's not going to be one institution will have enough samples or whatever to be able to study a given rare melanoma. So we will get back to that, but I'm going to have Keiran Smalley talk about his research.
All right. So I'd like to start off and reiterate some of the things our other panelists have already said. Firstly, huge thanks to MRA for actually helping support this. It's obviously a lot harder to have research on rarer cancers actually funded, and so I think having this kind of effort is extremely important. One other thing I'd like to actually say is kind of a shout out to all of you here, because we've actually worked a lot with advocates and patients, particularly those who have uveal melanoma, and I can tell you, you guys ask the best questions, actually. So I've had the opportunity to actually show around various groups of patients around our labs. They're happy to see eyeballs on slides and all kinds of hideous things, and they aren't like scared off by it and ask us some superb things, and I think it's been really inspirational. So, I mean, I will say that I'm primarily here to talk about our work in uveal. We do have an interest in acral as well, except I'll focus primarily on what we're doing in uveal. So really our interest in this came about quite a few years ago now, because we were kind of struck by the enormous progress that we had made in treating patients who had the regular cutaneous sun-exposed melanomas, because I've been up to this for like quite a long time now. So the time I started, the outlook for patients who had cutaneous melanoma had been incredibly bleak, and over a relatively short period of time, we're able to turn this around. We had a huge number of FDA approvals and really had increased the overall survival in patients. Not as much progress was obviously made in patients who had rare melanomas. As far as uveal is concerned, we didn't even have anything FDA approved until as recently as 3 years ago. In the past 3 years, we have, of course, had 2 FDA approvals, and I expect there'll be a few more happening soon. So I think that's really kind of indicating how much that having a kind of a focused program of research can really turn things around quickly. In terms of our interests, we actually have a number of interests in uveal. Our first one is really attempting to have a clearer understanding of why the high-risk patients who have class 2 uveal melanomas have metastases. So we're actually really focused on trying to understand some of the cellular programs which are activated in these class 2 tumors, which allow them to spread to and colonize other organs, particularly the liver. And then our other interest is really trying to understand the immune environment of uveal melanoma, because of course it's known it's not as immunogenic as cutaneous melanoma. There haven't been as strong responses to things like anti-CTLA-4 and anti-PD-1. We think that could be in part to the fact that uveal melanoma has a much lower tumor mutational burden. It's also probably some environmental factors. The eye is an immune privileged site, so is the liver. So it is probably a combination of understanding the immune environments of the host, like organ sites, as well as the tumor itself. And so those are probably our 2 strongest focuses on uveal at the moment. As far as acral is concerned, you already heard of Vito, that patients with acral melanoma are not responding as effectively to things such as immunotherapies. And so here again, we're applying some of the same techniques we're using in uveal to try and understand the immune environment and come up with new immunotherapy targets. And I'll stop there.
Great. Well, thank you for helping us understand what your research studies are. So Keiran just said some of the accomplishments and mentioned some of the accomplishments in terms of 2 approved therapies in uveal. Kasey and Vito, what do you think have been the major accomplishments in acral and mucosal melanomas over the last 5 to 10 years?
I can begin with acral. So the last 5 years or so in the acral melanoma community have been something of a renaissance. I mean, acral melanoma was first discovered in 1976, and then it went dark in terms of published reports in the literature. But over the last 5 years or so, we've begun to start shedding light in the mutational landscape, which has been shown to be almost, one would say, completely different from cutaneous melanoma. There's different driver mutations at different frequencies in acral versus cutaneous. There was a recent study just this past year from a team led by Boris Bastian showing that the sequence of mutational events is strikingly different between cutaneous and acral, essentially showing that it's a different disease altogether. There was a study from the Robert Judson Torres laboratory where they were looking at the cell of origin, the melanocyte, and how the identity of that melanocyte might be different in your foot versus sun-exposed sites. And so, of course, foot melanocytes are where acral melanomas are thought to come from. And when you look at their properties, they are completely different than melanocytes from sun-exposed sites, again suggesting that acral melanoma is a completely different disease. Those are really some of the key things that have been found recently in terms of potential drivers that can lead to the initiation. There was a prolific paper a few years ago from Richard White's team looking at zebrafish and finding that the fins and the tails of a zebrafish are very similar to the hands and the feet of humans. And using this approach to study acral melanoma initiation, he found what I think many would consider is the first potential bona fide driver of acral, this protein called crackle. And so, although not much has been done compared to cutaneous melanoma, the last 5 years have seen a renaissance of our first insights into what could be driving this disease. And I'm very hopeful that the next 2 to 3 years, significant therapeutic translation can potentially happen really because of all of these initial findings.
Kasey?
Yeah, so again, I think just because it's the rarest of the rare, the mucosal research, melanoma research field has really been lagging behind both uveal and acral. And early on, because as Joan showed in one of her slides, if you caught it in the pie charts, the genomics of mucosal are actually quite similar to acral. And so for some of the earlier publications, these mucosal melanomas were simply grouped with acral because there weren't enough mucosal melanoma tumors to really pull out that group and have it be a standalone group. However, really through collaboration and sharing of samples worldwide with groups, with the Australia group and Nick Hayward's group, groups at UCSF, we now have some publications come out really focusing on the genomics of mucosal melanoma and not just overall mucosal melanoma, but beginning to separate out the head and neck mucosal melanomas, for example, compared to the genitourinary mucosal melanomas. And so really, just within the last 5 years, I would say we finally have a good snapshot of the genomics of mucosal melanoma and what's going on there, and potentially learning some of the differences between the head and neck and genitourinary mucosal melanomas. We've also had, when I think of research, I think patient samples and analysis, and then moving to preclinical models and moving to clinical trials. So I think preclinical modeling has also really has had a lot of advancements for mucosal melanoma. So for example, there's been a couple groups now that have come out with zebrafish models and for mucosal melanoma to study. And another kind of interesting field is canine melanoma. And so dogs actually have a very high frequency of oral mucosal melanomas. And so there's been a really big surge in comparing these canine mucosal melanoma to human mucosal melanoma to determine, can we use canines as a model and perhaps even, run clinical trials in canines for mucosal melanoma specific therapeutic indications. And so I think the preclinical modeling has also really had a lot of advancements in the last 5 years. And then finally, mucosal melanoma a lot of times has been eliminated or excluded these patients from a lot of clinical trials. However, over the past 5 years, there have been a number of now mucosal melanoma specific clinical trials. We had one of our own, including anti-PD-1 in combination with the DNA methyltransferase inhibitors. Alex Shustari's has a mucosal specific trial at Memorial Sloan Kettering. And so I think really the recognition and the ability to have these mucosal melanoma specific clinical trials has been a huge accomplishment.
I just wanted to quickly add, in the acromelanoma field too, because I want to give credit where credit's due, Keiran Smalley's group also has been one of the first teams to do single-cell RNA-seq, a really cutting-edge approach on acromelanoma samples, really documenting some of the first characterization of the immune landscape, how we can find new targets to improve it. So I just wanted to also mention that.
Oh. And Keiran, you also, in terms of specific trials for the different rare subtypes, Dr. Hamid mentioned one of them, or at least the part I was in with Replimune. Keiran, you have an interesting trial going on at Moffitt in terms of testing a drug in uveal.
Which one in particular?
Okay, sorry.
So we have a number of trials.
Okay.
So again, historically, there hasn't been as much industry interest in such small groups of patients. But at a center such as ours, we actually see quite a lot of patients now. In part, this actually came about because one of our surgeons, Jonathan Zager, was instrumental behind the FDA approval of the Hepzato kit, which I think was a year ago. And because of that, we now see like a lot of patients who are coming to us for that. In terms of trials, we actually have 5 IITs open, which are exclusively for uveal at the moment, which I think is actually a large number for such a rare cancer. Some of these, off the top of my head, we have anti-PD-1 in combination with an inhibitor of the PARP protein. We also have one where we're combining a MEK inhibitor and a histone deacetylase inhibitor. We also have a very innovative trial, which is actually a targeted alpha particle, which interacts with a cell surface receptor, which is expressed on the uveal cells. And of interest, that project was originally an MRA funded project. It was actually a lab project we had at our institution, which has been actually opened as a phase 1 trial. And so I think particularly in the rare melanoma space, there's actually a lot of potential, I think, of having innovative ideas, which, for example, have been MRA funded, which are actually heading forward into the clinic at the same institution. And I think because it's so hard to actually move a lab idea forward into the clinic, I think it's extremely important to have supportive organizations such as MRA for this.
Yeah, thank you. And we were really pleased to see that MRA-supported project move forward into a clinical trial at your institution. So in the remaining few minutes, because I'm going to ask for questions in about 5 minutes, let's focus on collaboration and how we could think about collaborative efforts to really advance rare melanoma research. It'll take a village because, there's only a certain number of patients with these rare subtypes at each institution. So maybe I can have the panelists comment on, what kind, Kasey, you talked about your model collection, preclinical model collection, and how you share it with the community, and you are on our website, and I'm very pleased to hear, I think you said 40, you gave models to 40 labs. That's a great, that's great to hear. But what are some other ways that we could collaboratively work with institutions and also perhaps get patient input into what you're doing
No, that's
to
great.
advance research?
So yeah, I want to also, again, just reiterate what Joan said. Kasey has really brought back from the dead the mucosal research community by sharing really the only collection of mucosal PDXs that are broadly accessible and a lot of acral PDX. So Kasey really exemplifies this spirit of collaboration and sharing, and that's what's, I believe, really as a lightning bolt, increased the progress in these rare melanoma projects. So for our laboratory, something with acral is that it arises on different anatomic sites. It's most common on your foot, hand, fingernail beds, toenail beds. And so what has prevented the study of metastatic drivers has been the lack of a bona fide model of metastatic acral where you can put it, the tumor cells, into the same site that it arises in people. So putting these tumor cells into the foot. And so our laboratory over the last 4 years has optimized a metastatic approach where we can implant these tumor cells in the foot, and we're able to track them and characterize their changing biology when they colonize distant organs like the liver, brain, lungs. So this model took us about 4 years to create. There's some finickiness about it, some difficulties. And so one thing we've done to help the community is instead of everybody trying to reinvent the wheel, which we're happy to provide some, all the guidance to bring that model to their teams and their institutions, we've also provided in a collaborative fashion the ability to test whatever investigator ideas that come from outside of Johns Hopkins in our model system so that it saves them time, saves them money, and we can be sure to make the research progress as fast as possible. We've also created a few different cell lines that we share widely to different members in the community. For example, we've given some models to investigators at Cornell to make even more sophisticated approaches. So we make cell lines, they're able to make organoids, which allows the biology to be much more similar to the human instead of a plastic dish. But so these are just some examples of where collaboration can really make a difference by collaboratively helping each other with whatever superpowers and resources you have that are unique to your institution.
Yeah, and I just want to add one thing, that some of these models are not very easy to run, and they really require expertise like that these labs have. So I think it's great that instead of perhaps providing the models, a researcher could come to you and say, I have an interesting drug to test. Can we collaborate, and could you test it in your models? Because you would have the expertise in running the models, they have the idea and perhaps the drug, and this is a beautiful collaboration type of project.
I mean, I'd like to have a plea here to the advocates and the patients, is that we can learn so much by analyzing specimens as well. And so I realize this is a sensitive issue, except if you feel that you have specimens that you're able to share, these can be analyzed using all kinds of high-tech approaches. We can have a look at the immune environments, we can have a look for genetic alterations, and our plan and the whole reason that we have a sort of a collaborative community is after these have been analyzed, the information is shared amongst all of us. And so all of our groups can basically answer the same questions using the same few handful of specimens. And it's incredibly helpful as soon as these kind of omics approaches or the sequencing approaches have been happened, they can be analyzed again and again and again, and it is a great thing for all of us and all of you, because it really allows us to actually find kind of ideas and potential new therapeutic targets as well. And so I would urge everybody, if you have an opportunity, I would think seriously about this. It will help you, it will help us, it will help all of us.
Yeah, and I just want to add on one of my slides, we are creating a MRA melanoma biorepository where patients can be consented to donate either tissue that has been collected, or if you're going into surgery, prospectively collected after surgery, and it'll be stored at a central biorepository. And the purpose of this is to provide that tissue to researchers worldwide for research studies And also, what will be very important, and the researchers will have to guarantee, is that they share their data afterwards with the research community, so we could all benefit for it, and it's not just stored at one center. So Kasey, do you want to add? And then I think we'll have some questions.
Yeah, so I just want to, I guess, echo, what Keiran and Vito have said about the collaboration and tissue sharing and model sharing. It's really important. But there's also a lot of red tape that happens between universities, between institutions about actually sharing patient specimens. And it's no fault of the researchers that are there. It's a lot of the intellectual property, IRB. There's just a lot of issues with actually sharing the patient specimen. So I think it's really exciting what the MRA is doing by creating the central repository that could be essentially assessed by everyone. But I just want to finish by saying, I think with the rare melanoma patient community, it's not just the lack of rare melanoma cases, but it's the amount of tissue that we get on each case. And so I think, it's particularly important for us in the field to really be collaborative and work together and kind of know what each other's doing and really help each other, as well as, so we're not really repeating the same experiments and using the same tissues to do the same things over and over. These are really rare cases, but also usually rare amounts of tissue within these specimens. And so I think really making sure that we're collaborating and sharing what we can, whether it's derivatives of these specimens like RNA or gDNA or, or blocks. But I think it's really important that we're working collaboratively and not really, utilizing these specimens for the same, I guess, kind of overlapping purposes, if that makes sense.
Yeah, we call them very precious samples because they're from rare melanoma, melanoma subtypes, and they, so there aren't that many, and they're precious because they might be limited in the amount of tissue specimen there is. So being able to store them, work on them together, and really share the data is really essential. So I see we have about 5 minutes left, and I would welcome any questions that you might have from all of the participants in the audience. And I can't. Oh,
Yep.
okay.
I guess my question would be, I opted in to have my tissues examined. My question is, each biopsy I have, are those tested? Because I had one that came back as a c-KIT mutation. So are those continuously being tested as I get biopsied? Or is it just one specimen that is just checked, in a bank? Because each one for me has been different, each biopsy. So I guess that's just what do you do with all those different biopsies?
Yeah, so, the main part of the biopsy is going to go to pathology, and so then clinically, your tumor is going to be tested through the clinical mechanisms. In the repository, we bank everything, and then we do kind of a batch analysis. And so what we'll do is maybe once a year we go through and we'll run sequencing, RNA sequencing, a basic genomic platform on all of the tissues that we have. But in terms of, so it's not just you give one biopsy and then the others don't matter, because that's actually really important. And we, as a biorepository, will have requests for samples of a patient's biopsy before they went on immunotherapy and then when they're on immunotherapy. And so by having those multiple different biopsies at different time points, that's actually really valuable and for research purposes to actually use those. And so It's not immediately, as soon as we get in a tumor, everything is tested right away, but in terms of, for the research purposes, eventually we do kind of batch test everything. And then, again, we're collecting serum, we have blood genomic DNA, and so a lot of these materials are also getting sent out to investigators worldwide for their projects as well. And so, so the tumors, all of the specimens, the blood, the tumors, everything is really being highly utilized, not just by our group, but also by other investigators as well.
Yeah, being able to follow what we call longitudinally, so from perhaps the first biopsy and then across the disease is very useful to be analyzed for research purposes, to see how the tumors change or the immune microenvironment changes over the course of time and over the course of treatments. So I'm trying to look out and
Hi. So I have acral melanoma, and I'd be happy to donate a specimen, but I've been injected and biopsied a bunch of times already. Is it the sort of thing where if I'd want to donate something, I show up at your doorstep, or do I give my doctor, tell my doctor to mail you whatever tissue they have, or is it just too late, because they were all done in the past?
Well, we're gonna have FedEx show up at, not at your doorstep, but at wherever your specimens are housed. And so Joan could probably answer this a little bit better, but essentially it's gonna be a direct-to-patient repository. And so they're going to be, through a third party, Patty Cogswell and her company, identifying patients like you that have specimens that are already existing, and we're going to be shipping out kits and essentially getting that material, and then it'll come back to the biorepository central to be housed. So it'll be both existing tissues as well as prospectively for upcoming surgeries or upcoming things that'll be collected.
yes, so definitely stay tuned. We probably within the next few months, we will start posting on our website when the biorepository opens, that patients, if interested, can be contacted by a group that we're working with to consent them, and then once you consent, you will be able, they will be able to contact your doctors or surgical offices for any stored material, archived material, and also if anyone plans on going in for a surgery, can actually collect it as it comes out of the OR and get it shipped to the biorepository. So definitely stay tuned on our website, MRA website, in terms of when this announcement will be made. And thank you for wanting to do that. But we won't come to your door with UPS or FedEx.
Someone over there.
All right, we have time for one more, and then that'll be it.
Okay,
Yeah, there's
I
one.
can't.
Yep. It's hard to see.
I know you can't, it's
On a
blinding.
much lower level, when you're seeing a dermatologist and they send your biopsy out for that initial biopsy, I am thinking at this point that as a patient, it's maybe more beneficial for me to be more involved on that aspect of where that biopsy is going, where I have not ever thought about that until a couple of years ago. I had a biopsy and it was great, nothing came of it, and then I found out it was sent to California and it was not so great. I'm now a Moffitt patient, so I don't have to worry about that anymore. I'm a patient of Dr. Tarhini's. But do you think that as a patient, when we're seeing our dermatologists and they're sending things out, that that's an area of concern that we need to know where it's going? Or are there some labs that are just better than others?
So that's a little bit more of a clinical question. I guess a little bit less what we deal with on the research side of the biorepository. I would say, there's always challenging cases, and I feel like, these cases, every new case gets reviewed, and a lot of times the challenging cases are automatically sent out to a lot of experts at UCSF and other places. really to confirm whether or not it's melanoma. So, I guess in terms of tracking your own specimen and, making sure where it's going, I think that's going to be a little bit challenging. I think that's really something that you should be like discussing with your physician and be, I think you should feel open and able to have that conversation with your doctor. Where is my sample going? Are you sure that there is no cancer there? I think these are really things that you should be, not feel bad to really ask your physician and have that relationship with your physician.
Now it's great because I'm in a big institution. I have no worries about it anymore, like none. But before, I just didn't think about it.
I will actually add the kind of samples that tend to come to us as researchers are usually pretty far along in the process. So we already know that you have like a rare melanoma and then you're having a surgery, and at that point you should be like approached and asked. It's like, can we have a sample of this for like research purposes? I think the question actually you had was at that kind of earliest point, and at that kind of point we are not usually involved at all. So
But I'll end by saying to all the patients and advocates in this room, please feel free and all the time ask your doctors questions. Like if they're doing a biopsy, why are you doing it? Where is it going? What information will you get? It's so important to advocate for yourselves and make sure you understand this information. And I'll leave it at that. And thank you so much. I want to thank the panelists, who I enjoy working with immensely, and just enjoy the rest of the forum.